Hello, friends. Welcome to Science Talk. What follows now is part 2 of plastics in the microbiome, picking up with mitigation. Here we go. Mitigation of plastic pollution by the gut microbiome. Recently, several insect species, as example, larvae of darkling beetles, wax moths, meal moths, have garnered interest for their ability to consume and degrade a diversity of plastic polymers. That's the important part, degrade. For example, larvae of the Indian meal moth Plodia interpunctella can ingest and appear capable of degrading polystyrene, as do larvae of yellow and giant mealworms, Tenebrio molitor, Zophrobas moria, respectively, larvae of the greater and lesser wax moths, Galeria melonella and Acroia gracilla, are similarly reported to degrade polyethylene and polystyrene, respectively. Isotope analysis provides evidence that carbon from plastics, such as PE, is incorporated into the biomass of invertebrates. And, they're not quite sure what effect this had, but it's happening. So that is of concern. Let me see if I can enlarge this one again so we can look at this. Evidence for role in plastic degradation by higher organism. Basically, eukaryotic multicellular organism. A survival of germ-free insect larvae on plastic as the sole food source by gut microbiota. So we got increased microbial abundance in the gut. Disruption of the microbiome, for example, taking antibiotics, reduces the plastic losses, which sounds like a good thing. You get rid of the plastics from your body because when you take the antibiotics, you're killing bacteria. You may be killing bacteria that are utilizing the plastic. But if the taxa are breaking down the plastic, you probably don't want to remove those from your body. And isolation putative plastic degrading microbes from gut and frass. I don't know what frass is, to be honest. And then we have gut-independent microbes, growth of isolated colonies on plastic as the sole carbon source. And then in the little description here, evidence for role of insects, host-associated microbes, host-independent free-living microbes in plastic degradation. Degradation of the plastic polymer may be detected by a variety of methods, including mass loss of plastic, such as clear zone development around the colonies on plastic-infused overlaid agar, altered plastic surface properties, generation of the degradation products, for example, CO2, polymer metabolites detected by Fourier transform, infrared spectroscopy, or high-performance liquid chromatography. Many organisms consume plastic incidentally and gain no nutritional value from its consumption. Plastic has been found in abundance within the guts of diverse organisms, from seabirds and fish to marine and freshwater worms and zooplankton. Although the ingestion of plastic by species, including the common earthworm, is associated with reductions in plastic size distribution, in many cases, demonstration of plastic degradation, for example, conversion of CO2, incorporation of plastic-associated carbon into animal biomass, is unsubstantiated. Similarly, the consumption of plastics by mealworms and wax moth larvae has gained much attention. But confirmation of plastic degradation by the host gut-derived enzyme, independent of host microbiome, requires further confirmation. So it's unsure yet as to what's happening, if that's a potential way to remove such pathogens. And they talk about using radioactive tracers, C13, C14, to see what's happening, what the fate of everything is. And they talk about how some of the microbial taxa that appear in, like, say, the digestive system have been identified through various means and so forth. Prior to the study of Casson et al., multiple authors have already isolated putative plastic-degrading bacteria from the insect gut microbiome. Yang et al. Isolated the bacterium Exiguobacterium species from the gut of styrofoam-fed mealworms and demonstrated its ability to grow on polystyrene film as a sole carbon source, typically associated with changes in surface topography and hydrophobicity of the plastic. Basically, hydrophobia, a fear of water. The first evidence that free-living environmental taxa contribute to plastic degradation was only published about 30 years ago, circa means about, after the first commercial plastic production. Excuse me. In 1974, and Fields et al. Showed that the fungus Oreobasidium pullulans was capable of PCl degradation. Since then, the number of microorganisms suggested as capable of plastic biodegradation has increased considerably. A recent study by Gambarini et al. Reports over 400 publications describing the degradation of 72 different plastic types by 436 species of fungi and bacteria. Presumptive plastic degrading microbes identified to date belong to 5 bacterial, 3 fungal phyla. And then among the bacterial phyla, we have proteobacteria, actinobacteria, firmicutes, and bacteroidetes, and cyanobacteria have far fewer. The fungal phyla include Ascomycota, Basidiomycota, and Mucoromycota. They kind of show the grouping here of what I just tried to pronounce. As outlined earlier, a small number of plastic-degrading microbes have been isolated from plant and animal-associated microbiome. However, most isolates reported in the literature were derived from soil or from waste processing sites such as composting facilities and landfills. And an additional source comprises bacteria and fungi already deposited in culture collections. All major synthetic polymers have species reported to degrade them. Bioprospecting for novel mechanisms of plastic degradation. In other words, looking for ways to break down the plastics. Now, the key is you want to break it down, but you don't want to simply just break it down to the point where you just simply have, individual molecule. You want to break down the molecules so that you can release elements, carbon, oxygen, nitrogen, that kind of stuff there. That's how you really get to removing this from the environment. Because if you just break it down to smaller pieces, but you still have the functional groups intact, and all you've done is create nanoplastics, the issue still persists. The problem still persists. So keep that in mind as well. You want to actually break down the molecules. There is a lack of information necessary to critically validate many reports of plastic degradation by microbial taxa or communities, or to accurately reproduce the research. Okay, this is troubling to me. Reproduce the research. When you write a scientific paper, you include a methodology section. The purpose of the methodology is, several goals there. One is, so that the reviewers, the reader, can assess how good the experiment was done. How well was the research done? What is the mathematics sound? Is the methodology sound? Are the statistical analyses sound? Etc. Etc. Another aspect is that if another researcher wants to be able to do the research that he or she is reading, they should be able to take the methodology and be able to set up the experiment the exact same way as those that just published it. And if you can't actually reproduce those results there, that to me is troubling. And they give some examples as illustrating that aspect. And figure 4, okay, we'll go large size here. Okay, keep in mind, folks. Okay, I'm basically a physical oceanographer, so this is outside my expertise area here. But plastic is a major problem. This is why I'm bringing this report to you. So for those of you who may know something about tensiometry or viscometry and so on, you can then assess this paper. There is an extensive literature review at the end of this paper, so you can pull up those papers. You find something that interests you, you want to read more on it, it's there. So I'm going to leave you the URL. And then so they have methods used to assess changes in polymer structure. So they have changes in polymer structure in the blue, nice little Venn diagram. Plastic degradation, methods used to assess plastic degradation, and then methods used to assess generations of plastic metabolites in green here. And then you have this overlap. Okay? Percentage of studies using evidence for plastic degradation by microbial species based on changes in polymer structure in blue, physical loss of plastic mass in red, it looks more like pink to me, detection of plastic metabolites in green, or in combination. Basically, 24%, 20. So if you look at where all three intersect, it's about 10%. Most reports, and this is important information that's needed to be ascertained. Most reports of plastic degradation by microbial isolates do not go on to explore the genes and enzymes responsible for that activity. I think that's critical information. If you at least know the enzymes, you can then do gene manipulations so that the microbe produces those enzymes, and then you can then proceed to break down the materials. So I consider that to be important information that hopefully more of that type of research will begin. In fact, only 14% of the microorganisms reported to degrade plastic have the gene sequences conferring the degradation activity elucidated. This represents a major shortcoming since knowledge of the relevant biochemical molecular data provides the capability to advance the plastic biodegradation field enormously. That makes intuitive sense, I'm sure. I have a table here. Best practices for reporting microbial plastic degradation. We describe info, techniques, and practices critical to get this information. Let's take a look. And so you have the item, plastic identity, composition, microbial taxonomic classification, isolation, environment and conditions, strain accessibility, assessment of plastic degradation, plastic degrading enzyme and gene identification. They give you description, importance, and best practice example, which is really a literature review. Okay. In other words, look up the sources here to get the further information. So, descriptive name for the polymer, the molecular weight, the source, studied reproducibility, complete polymer composition, differentiate the biodegradation of the polymer and the additive, taxonomic classification, reproducibility, extrapolation of findings to related species. Look at the phylogenetic distributions, strain, isolation, location, site-specific properties such as temperature and pH, so you identify the environments favorable for degrading plastic. Deposition, description of isolated strains in international culture banks permits greater reproducibility, further study by other researchers. Description of techniques used for confirmation of degradation. Okay, so you confirm the degradation. It's important to differentiate, or it's important to confirm how the techniques differentiate between degradation of the polymer and additive where included. And then identification of the enzyme responsible for the biological degradation and its gene sequences, allows the mining the molecular database, recombinant gene expression, enzyme optimization, etc. This to me is almost sounds like the key point here. If I identify the enzymes that break down the stuff, that's what you want to enhance and figure out a way to basically mass produce that. Now here's an interesting statement here. Based on protein mutagenic and structural analysis studies, alongside homology database searches, it is likely that certain microorganisms already possess plastic degradation genes but do not express them in situ and or derive energy from more readily utilizable carbon sources when available. So it's almost like they have the potential, we just need to turn it on. By incorporating inert controls, for example, a glass ceramic surface, we may be able to distinguish between genes acquired and expressed for the process of the plastic degradation from those normally expressed in biofilm communities. In other words, including where plastic is not present. Okay, mere changes in polymer mechanical properties and physical structure, even when observed in concert with microbial biomass production, are insufficient evidence to confirm polymer biomineralization by microbial isolates. Physical losses of plastic mass should also be reported. Plastics can be incorporated into growth media as plastic film, powders, or granules emulsifications. Here, a common method for assessing microbial plastic metabolism is by observing clear zones in agar containing emulsified plastics. Now you can see a clear zone, some sort of digestive, some sort of chemical process took place there to create that clear area. Knowledge of genes known to be associated with plastic degradation provides a strong tool to identify new degraders and genes among microbiome communities. And we go on to cite a whole bunch of works here of people doing that. Manipulating microbiomes to enhance rate and extent of plastic degradation. Notice make it on a larger scale. So you can employ different strategies to overcome the challenges of isolating the microorganisms that can do this sort of stuff. Maybe higher temperatures increase the flexibility of both amorphous and crystalline domains of the polymer chain, thereby improving the accessibility to enzymatic attack. That's basically setting them up to be broken down. In this regard, thermophile microbiomes represent a promising source of enzymes because they'll likely be more thermal stable. Thermal temperature file loving. So these are microbiomes, microorganisms that love it warm. Okay? Just like, acetophiles, they like it acidic. Degradation rates will further increase after improving enzyme thermal stability through site-specific mutagenesis, basically gene manipulation. To date, however, only 10% of isolated plastic degradation studies report polymer degradation at temperatures greater than or equal to 50 degrees C. Only a small fraction, about 0.5%, of these have been isolated from extreme environments such as hot spring, compost, anaerobic digesters. There would appear to be significant scope for mining thermophile, extremophile microbiome as a promising source of putative plastic degrading enzymes and microorganisms. And by the way, when you see numbers like 186, 192, that tells you that there's been at least 192 papers cited, papers used in putting this paper together. This is a big literature review is what this is. The higher genotypic and phenotypic diversity present in microbial communities compared with single microbial strains may mean that communities are more efficient degraders of xenobiotic pollutants. As such, artificial consortia created by selecting a small number of plastic degrading microorganisms within an already existing consortium, top-down approach, may be a useful strategy for improving plastic biodegradation. Alternatively, directed mutagenesis to improve gene expression and enzyme function, along with metabolic engineering and synthetic biology tools, could be exploited to obtain more efficient plastic degrading consortia. Specifically, the introduction of modification of interspecific microbial interaction, such as intercellular communication via metabolite exchange, could be used to create consortia with improved biodegradation traits. Additionally, segmentation of metabolic pathways among strains such that each organism produces an intermediate compound that can be used by the next organism in the pathway can be used to reduce the metabolic burden on any one organism. Like, okay, I've done my job. I'm handing off I've run my leg of the relay, so I'm handing off the baton, basically. Basically, only limited information is available regarding genes and enzymes involved in plastic biodegradation. An improved understanding of the degradation pathways by single strains and multi-strain co-degradation pathways is first required to facilitate this approach. And it brings you to the conclusions here. Impacts of global plastic pollution. Microbiomes are diverse, ranging from direct consequences of toxic leachates on microbial community health and activity to the indirect effects of plastic on host organisms and environments. Many hundreds of microbial species, genes, and enzymes are implicated in plastic degradation. For a small number of particularly bio-based plastics, such as PLA, clear evidence is presented for their microbial degradation. However, for the majority of commercial plastics, evidence for microbial degradation remains weak, with studies failing to confirm microbial growth on the synthetic polymer. Ensure the correct identification of plastic degrading taxa and enzymes, facilitating their improvement by environmental, biotic, genetic manipulation. Multiple lines of evidence for plastic degradation should be presented. Ideally, this will include evidence of changes in the polymer structure, mass loss, detection of degradation products, along with confirmation of the microbial strain and putative plastic degrading enzymes and associated genes. Such details are essential for organisms and enzymes capable of plastic degradation to be reliably differentiated from those only capable of degrading the more labile carbon within predominantly amorphous plastics, plastic monomers, fillers, additives. And here's a list of abbreviations. There's a wee bit there. PLA, polylactic acid, we've been discussing. And so still give you a bunch of plastics abbreviations there. And here are your references. And it continues. So let's see, how many papers did they cite in here total? I saw it was up to 192. So they cited 196 papers. So you can look at a Google Scholar depending on what you have access to. A lot of these papers are going to, unless you subscribe to the journal, you may be able to pull up the abstract, but if you want to see the details of the paper, it's probably going to be behind a paywall. So keep that in mind as an example. So basically, and then they tell you what funded the work and so on. So basically, what they're doing here is they're identifying all the problems that plastics presenting, whether it's to microbiome communities, environmental microbiome, gut microbiome. We touched upon some of the deleterious impacts the plastics have on organisms. Killing them. That's kind of bad. Or just the chronic situations. Creating obesity, or on the other side, weight loss, creating issues where it compromises the immune system, and therefore leaving the person more susceptible to infection or some sort of a disease, what have you. and then we actually ingest it. It gets into our tissues. That causes other issues as well. So they talked about all these problems. I mentioned the organisms eating, the stomach's full of plastic, and now they're basically saying what are the ways to mitigate this? What needs to be done? Here are the things that should be looked at. Here are the methodologies that should be explored, should be carried out in experiments to find out what works best. But you got to keep certain caveats in mind. So this is a literature review, but I've been bringing to you from time to time stories involving plastics. And this is just me continuing to do so, and you can this is just published this year, so this brings you the latest knowledge, as fact that they cited 196 papers. This brings you the latest knowledge on the situation of basically microplastics. So I said I will leave the URL link in the comments section, and those interested, you can read the details at your own leisure and your own convenience as well. So until next time, thank you for your time. Hello, folks. This is Jim here with Science Talk, asking you to please subscribe to my channel and to inform others of my channel and of the work that I do. Please share to social media platforms that you use. Also, as a reminder, don't forget to click the bell so that you know when I load up more videos. Finally, I ask that you support the work that I do by becoming a patron at patreon.com. Details in the description box below. Thank you for your support.